All right, everybody, we're going to try this again. Please let us know if the buffering is still an issue. Fingers are crossed that it is not. So I'd love to try this. It was just a dry run of our intro. It's always good to practice. We could try it again. Try it again, try it again, try it again. Oh, so smooth. New stream. There we go. That's it. That's it. The internet version of turning it off, trying it over again, toggling the plug, so to speak. So let's hope that everything works here. Let's see. Let me tweet to everyone. Hey, TWIS. Let's see if I can type. On. I know this is a really compelling video at the moment. Okay. Oh, and of course, I didn't tweet it to the right places, but it's out there. It's in the wild. It's going there. You ready to start a show?
Let's do it.
All right, we are going to make this happen now. Starting in 3, 2. This is TWIS. This Week in Science, episode number 678, recorded on Wednesday, July 11th, 2018. The Science Drive. Hey everyone, I am Dr. Kiki, and tonight on This Week in Science, we are going to fill your head with an ancient Chinese secret, American dogs, and snorting horses. But first,
disclaimer. The following program has been constructed entirely out of scientific materials. No corporate industry, government ideology, philosophy, religion, or special interest lobbying effort was involved in its creation. Sponsorship of the show is generated by a community of its listeners who have no control over the content of the show. The hosts themselves have only the vaguest recollection of volunteering to be hosts, and were not selected to do so by anyone other than themselves. None of them need your vote, your money, your trust, or your time, and you will not need to click agree to any form of end-user agreement in order to enjoy the content. The stories you will hear are real accounts from the publications of lab and field notes of scientists engaged in research and discovery. The only filters between you and the subject matters discussed are the education of the experts generating the information and the cadences of the host's delivery in relating that information to you. If at any point while listening, you begin to think too freely, feel free to find a more conventional format of public consumption upon which to dull your senses. We have no time for that sort of nonsense here on This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
good science to you too, Justin. And we are without a Blair this evening. She is off sciencing in the name of the climate somewhere. I think she's on the opposite side of the country. She probably used a lot of carbons to get there. But anyway, enough of that. She'll be back next week. In the meantime, Justin and I are here to talk all about the science that's happened for, I don't know, the last 2 weeks, because we took a vacation, right? We had like a night off.
Yep.
Did you enjoy your 4th of July?
I did.
Good. For me, it was more than a night off because additionally, I didn't have to edit or publish or do anything on Thursday. So it was like 2 days off. It's amazing. I was like, wow, it's like a whole week off here. This is amazing. But we're back. I missed this. I'm glad. I'm glad to be back talking about science. What do I have? What do I have on the show this week? I have stories about looking for dark matter, new ideas in the search for dark matter. Also, I have a story about dogs in America. That's right. It's exciting news. And I actually put that that story up because I think Blair would have brought it to talk about the dogs in America.
Yeah.
And CRISPR for gene drives because CRISPR.
Yeah.
What do you have?
I've got an ancient Chinese secret, new, older Native American find, and multicultural African roots, as well as how to get a better brain in 15 minutes, and because Blair isn't here, snorting horses.
I don't want to snort horses. Doesn't sound like fun at all. Okay. The newest fad, snorting horses. Just don't do it, people. Before we jump into the show, I want to remind everyone that you can subscribe to the TWIS podcast if you have not done so already, or if you know somebody who would appreciate this, they can subscribe too. Easiest way to do it is visit twis.org to find information, but we are available all places where podcasts are found. But now it's time for the science. So, diving in, I would like to jump into my big story for this week, which is on gene drives. And the idea the researchers have put forward is a description of the first ever successful implementation of a gene drive in mammals. Now, here, let's answer some questions. Gene drive, what is it? Well, it's basically the idea of the selfish gene put into practice. It is a concept in which a gene does not get weeded out over time, does not lose its frequency in a population, but actually increases in its or maintains its frequency in a population. So, for instance, the idea of a gene drive, we could insert a gene into the genome of a mosquito that would reduce their ability to reproduce. And over time, the gene would drive itself through the population, basically leading to the death of mosquitoes or massively reduced mosquito populations because the gene reducing their ability to reproduce would keep them from reproducing over time. Yeah. This is the implementation that we've been thinking of. Other dreams that researchers have are usually in the scope of biological control. So, for instance, using them on mammalian pest species to bring down their numbers in the wild in a natural way that potentially won't have much impact on other species. However, there are concerns about gene drives going out of control, about the species intermingling, as we know species are wont to do, and for genes like this to go out of our human control and go out of the bounds of what we originally wanted and to become detrimental to nature as a whole. In any case, go ahead.
Which eventually, if you think about it, is inevitable in a gene drive. Well, I mean, for whatever species has it or it goes into, right? So, as if, for instance, there was genes in our ancient hominin history that probably had us be a lot hairier than we are today. And if that was in there like a gene drive, everyone would be hairy, even to this day. There would be no unhairy people. Right?
And
like
we wouldn't have stories about skunk apes and Bigfoot. It would be stories about the naked man.
There was a man with no hair seen in the mountains.
So strange. So very strange.
Yeah.
Yeah, indeed.
A trait that just never leaves the population. At some point, most of our traits, we can move on from a lot of them. Some of it is fundamental, but some of it,
yeah, and there are some genes that naturally have a drive to be maintained because they are really beneficial, and so they are within areas of the chromosome that don't get swapped and don't get changed out a lot during meiosis, during a part of cell division in which DNA does get swapped from between different areas of chromosomes. And there are other genes that, because of their positioning and where they are, they do get maintained over time. They don't have a lot of mutations. They're pretty well maintained. But most genes are going to undergo some amount of change over time, and their frequency in populations does adjust as a result of environmental influences, survival, adaptation, heredity, all these things are involved. So anyway, the interesting aspect of this research, not the cause for concern aspect of this research that I have brought up, but another interesting use, not just in biological control situations, is in the development of strains of mice for laboratory use. Last year, we spoke with a representative from the Jackson Laboratory, and the Jackson Laboratory is responsible for creating strains of mice that researchers use to do their research. And very often, these strains of mice have one gene that's changed in them, or one gene that's added, knocked in, or knocked out, taken out, made less useful genetically. And the problem is most traits are not based on a single gene. Most inherited disorders are multi-genetic. They have lots of genes that are involved in influencing how they work and how the disorder works. And so researchers are now asking for mice with 5, 6 different genes changed. And it takes years to develop these strains of mice. Imagine if with CRISPR or other technology, we could create a gene drive to increase the number of genes that are manipulated within a strain of mice for laboratory research use, and it could happen much more quickly.
And one of the things I thought was unintuitive was that it wasn't as simple, this whole knocking out a gene and then breeding a next generation of lab mice without this gene. Because as we were described by the Jackson Lab folks, you could knock out the same gene in the mother and the father and their offspring. Some of them will have that gene back again, even if it was knocked out. Like it's a tricky thing. And so they have to do all this, even the breeding program after they've done the gene knockouts or additions isn't straightforward. There's still quite a bit of failure that takes place along the way. So yeah, if you had gene drive that assured that these genes would be passed on within this population, it would definitely help and accelerate that process.
Absolutely. And so these researchers published this gene drive study. They have posted it on the bioRxiv, which is an online site for preprints so that they can get comments from the research community before it goes to actual publication. It has now actually been submitted for peer review in a journal, and the researchers themselves are not speaking with the media yet, according to Science Magazine. But the study is available on the bioRxiv. And what they did, the description of what they did is actually quite interesting. So they used the CRISPR-Cas9 system. But in what they did, they put the Cas9 enzyme, this is the enzyme that cuts DNA and snips the DNA so that the DNA can either have a gene stuck in it or taken out. They put that on one of the chromosomes in females. So they engineered female mice to carry Cas9 in their genome. Then they engineered male mice to carry the guide RNA that takes the Cas9 to the target that it's supposed to cut in their genome. And they added in a gene for coat color. So when the female and the male chromosomes combine during the reproductive process, oh, Cas9 is there, and so is the guide RNA along with the coat color molecule that will be slipped into the offspring's genome.
Wow.
Yeah. And so separately, it doesn't work. But when they reproduce is when it becomes interesting. And so the issue here, though, is that they found that the experiment did not work in males. And it's a result of how the cellular division process happens in males. Spermatogonia go through a mitotic cell division before meiosis, which gets in the way of a process that they're trying to take advantage of that's called homology directed repair, which is where when Cas9 goes in and cuts the DNA strand, the homology directed repair is the process by which another gene can be slipped in there and the DNA put back together in an easy way. And that's what they wanted to have happen. But it didn't happen in the males. In the females, the gene drive worked. So female offspring, it's copied the coat color modifying gene to the partner chromosome in lots of eggs. And in one mouse, 79% of her eggs ended up with the coat modifying gene on both chromosomes. And if she mated with a male without the gene, about 90% of her pups went on to inherit the gene. And they have this system of active genetic elements that they think could speed the creation of mice that have introduced or crippled genes, depending on what they want. The optimism, they say, and concern that gene drives may soon be used to reduce invasive rodent populations in the wild, though, is likely premature because they found that because the Cas9 and the guide RNA are on different chromosomes, naturally in the wild, they would become separated and not stay together as is needed for the process to move forward. So it's got a little bit of biological control involved in just how they set it up to begin with. Yeah, but gene drives, never before demonstrated in mammals, always in fruit flies and other insects, in mosquitoes in the laboratory. But this is the first report of gene drive in mammals. So who knows? This could lead to some very interesting work moving forward.
Yeah.
And CRISPR once again, which is very exciting. And then in other aspects of CRISPR, we've talked a bit about how CRISPR is inefficient, how it doesn't always edit the DNA in the right place. It doesn't always get it right. It has what's called off-target effects. And in a study published in the journal Molecular Cell, researchers just showed that when gene editing using CRISPR fails, and that's about 15% of the time, it's due to persistent binding of the Cas9 protein to the DNA at the cut site. And that Cas9 kind of gets stuck to the place that it was cutting. And because it's there, it blocks the DNA repair enzymes from getting in there and putting the cut back together. So part of the problem with the system not working is that the Cas9 just gets in the way. It gets stuck. And so they have to figure out, if they can figure out how to shake the Cas9 loose, it can potentially lead to improved efficiency.
Yeah, that shouldn't be too hard. That should be pretty straightforward. I mean, I think if I'm getting this right, they just are just noticing that this is why a failure is happening at times.
Yeah, it doesn't explain all the failures, but this is one aspect of the failures, right. And it gives them, if you have a dud occurring, how can you fix that? How can you get rid of those moments?
It's an enzyme though. You should be able to change temperature or pH or something and get it to fold itself slightly differently. That might be enough to shake it loose. Yeah, be interesting to see how they solve this problem.
Yeah, it has to do with the Cas9 interacting with the DNA strand. And then how do you get the RNA polymerase to get in there after the fact? Anyway, they'll work on it. What do you have?
That's a good question. What do I? Ancient Chinese secret, or rather, ancient tools and bones discovered in China by archaeologists. They are revealing that humans-ish, ancient human-looking hominins, at least, were in Asia much earlier than previously understood. So, this is much earlier. This is around 2 million years earlier, to be somewhat precise. They were found by a Chinese team. Well, not 2 million years. 2 million years ago, but not 2 million years earlier. They were found by a Chinese team that was led by Professor Zhao Zhu of the Chinese Academy of Sciences, including Professor Robin Dennell of Exeter University. The tools that they discovered were discovered at a locality called Shengchen in the southern Chinese Loess Plateau. Loess is a word somewhere in some lexicon meaning wind-blown dust plateau. The oldest tools that they found were dated at 2.12 million years old, which is 270,000 years older than what was the oldest skeletal remains and stone tools, which were found in Georgia. So this was previously the earliest evidence of humanity outside of Africa, and just got bested by 270,000 years. And
That's
at that
crazy.
site, they found enough skull fragments to reveal a human or hominin that looked like a cross between different hominids. I think they ended up calling it a Homo erectus, but they it looks, kind of Neanderthal-y. It's got some. I will call it that because Homo erectus can be diverse, I guess. All right. That was the one in Georgia. At the current site in China, the bone fragments are far too scarce to draw really big conclusions. They did find jawbones. There are some teeth. These may, upon analysis, give some clues. The tools they found were they're stone tools, scrapers, hammer stones, pointed pieces. They all show signs of use. The stone tools had been intentionally flaked. Most were made of quartz-like materials that came from the nearby mountains and streams. And fragments of animal bones were also in the 2.12 million year old range. So, yeah. This is an area, this plateau, I guess, is named Windblown Dust Plateau for a reason. It's been dumping dust there to the height of about 300 meters over the past 2 million years. So you could sort of picture whatever was there in the ancient past keeps getting layers of dust blown across over it. So there's got blanket after blanket after blanket, which in a way is preserving or at least protecting it from normal erosion. And so this is, of course, now going to be a fantastic place to look, especially now that they found something. 80 stone artifacts were found in 11 different layers of fossil soils. And they could determine that those 11 layers were developed in a warm and wet climate. Further, 16 items were found in 6 layers that were developed under colder, drier conditions. The 17 layers together were formed in a period spanning about 1 million years. So this shows that the plateau had this hominin habitation on and off again over 1 million years of history.
Wow.
They used magnetic properties, the pole flipping. They matched that up with other areas where they have good data for that to determine some of the times. Professor Dennell says, our discovery means it's now necessary to reconsider the timing of when early humans left Africa. And by early humans, he means really that if we're saying humans and we mean us, it's before humans. But if we mean humans is all Homo hominids, then yeah, that's early. This is a quote from Michael Petraglia, Max Planck Institute paleoanthropologist. He says it's entirely possible that Homo erectus occupied China at this time, but given the age of the site and the possibility that artifacts may be found at even earlier ages, another member of the genus Homo may be occupying Asia, such as a Homo habilis-like ancestor. So, and then Maria Martinon Torres, director of Spain's something to do with this archaeology, suggests that even some Chinese found hominin fossils once labeled Homo erectus are worth revisiting now. Again, Homo erectus has been given a pretty wide berth of morphologies because that's the only name they had for really old hominids outside of Africa, right? Like this is the one. So they just evolved a lot of different skulls, right? Well, maybe not. I mean, more and more we understand about this braided stream. These were compatible creatures with whether they were separated evolutionarily by 400,000 years or a million years. They may have been able to reproduce. So we may be looking at a lot of Homo erectus, maybe hybridizations of Homo erectus that we classically sort of identify and other early hominins that were wandering around. It's time to accept that not all hominins found in Asia fit in as the Asian Homo erectus taxon, a species that has been largely employed as a blanket term, says Maria Martinon Torres. I think that the question about the first Asian hominin identity is not yet closed.
No, the identity is absolutely not yet closed.
No, it's being opened up. Right?
It's being this is the initiation of it, really, of this really big conversation happening of, okay, there were multiple waves out of Africa, and there was obviously one very early, and these peoples, adapted and evolved separately from their relatives on the African plateau, on the African plains. Yeah, so who knows? I mean, maybe this is even another branch, multiple branches.
Right. I mean, we look at hominins getting all the way to Indonesia. That was so far dating back to about 700,000 years, right?
Right, but this is like over 1 million years ago. Right. Almost, this is 2 million years ago.
Yeah, this beats Neanderthals by more than 1.5 million years.
Yeah.
So this is tools. This is cutting tools. These are not just a stone tool. This is a variation of adapted tools for different uses. So very fascinating. One of the reasons so much of our understanding of early hominins coming from Africa is that we've looked there more than anywhere else, and we've looked mostly in a sweet spot in Africa, where it's relatively easy as compared to other environments to find fossils. And I've got another story in the second half that actually goes into, forget all of the out of Africa early cousins that left, but there's a new study out looking at the evolution within Africa of what is our current species of the modern human, or mostly, right? We got a little bit of braided stream in some of us as well, but it paints a picture of Africa of not being one population. And again, this Rift Valley was the one population for a long time. Again, a lot of research was done there. If you were going to be an archaeologist, paleoanthropologist, you went there to go look for bones because that's where you find them. So everybody looked there, and that became the population. But within Africa, there, which is the story in the second half,
many places,
so many distinct, separated populations by vast periods of time that would sort of get back together and separate and get back together. So our, the whole story has more chapters in it. But you have these Homo erectus type. Maybe, like you said, maybe it's something that we just haven't identified yet. Millions and millions of years ago, And then you have to start to wonder. This is a puzzling thing, because what if this is actually more of an ancestor for Denisovans or Neanderthals even?
Yeah.
And it changes the Denisovan and Neanderthal out of Africa story a little bit, because it might mean it's quite possible that they could have evolved on the Eurasian continent. If we now know that there's ancient forebears there already.
Yeah. From whence did they really come? Yeah. Everyone originally came from Africa, but there was little tribes that headed out and started whole new lineages, possibly. Yeah.
Well, the modern, yeah. I think we there's a lot going to end up being a lot more to this story. I think it's now possible. It's now possible, at least, that, say, a Denisovan or a Neanderthal really did come from Eurasia in terms of its origin story. So we may be tapping into origin stories of other distant cousins here too.
Yeah. So interesting. Tapping into origin stories, that is something that researchers are trying to do in many different areas of science. And in the area of dark matter science, researchers have a new idea on how to search for evidence of dark matter. We have ideas of what might make up dark matter. Some people think they're these weakly interacting massive particles, or WIMPs. And there are experiments that can potentially be designed based on what we think dark matter is made up of. And so Catherine Fries, who is a physicist at the University of Michigan in Ann Arbor, and her colleagues are suggesting that there are minerals deep inside the earth that, if we can access them, which might not be that hard to do, since we already do core drilling for other scientific experiments. You can there are earth cores taken out all over the place. If we can access these minerals like halite, which is sodium chloride, and zabulite, or lithium carbonate, that they are detectors within the earth, and that their nuclei, when hit or rung like a bell by these WIMPs, should have very specific markers that are left within the mineral itself that could be detected.
Wow.
Yes, they haven't done any of this yet. It's just a proposal for an idea. They're saying, okay, let's take a look at this. And so what they're hoping to do is get this after effect of the collisions of these particles, dark matter particles, with the nuclei of these atoms. And they're suggesting that the collisions may give us evidence that other experiments that we've created, that physicists have built, have not given us. They argue that halite and zabulite they're already deep inside the earth, and because of being deep inside the earth, in where they're formed, that shields them, insulates them from the effects of cosmic rays. So you can't just go grabbing rocks from the surface of the earth, because cosmic rays are bouncing all around and hitting the surface of the earth. However, dark matter particles can head right through because they're weakly interacting. They don't hit very many things. And if they smashed into the nucleus of sodium or chlorine, it would cause a collision and a recoil of that atom, and it could etch a path anywhere from 1 to 1,000 nanometers long within the material. And so they could extract these potentially 500 million year old minerals from boreholes that are already being used for oil and geological research, crack them open and look at them under electron or atomic force microscopes for these evidence tracks. Could also use X-rays or ultraviolet 3D scanners to look at things. However, there is a question as to whether or not something like natural radiative materials like uranium or thorium might be interacting with these sodium and chloride atoms and their nuclei under the earth as well. And so they'd have to figure out what a background signal from the earth's own radiation would look like compared to these tracks from the recoil of the collision, the collision detritus within the atoms themselves. But it's a new idea.
And I guess you'd have to bring it up in like a lead box and make sure that, like, you know, but whatever. Yeah. Like, yeah, this is a fantastic, at least excuse, not excuse, but reason to go and investigate.
Absolutely. And I love the quote. It's in this Nature article from Freese herself. And she adds, it might be possible to identify these etching patterns created by bursts of radioactivity and to ignore them. But the truth is, you have to do it and find out.
Yeah. Mm-hmm.
You know, you do. You have to do it. And so go for it, Dr. Freese. Go figure it out. I want to hear the results of this idea. What else do you have there, Justin? Tell me another story.
I got more older ancient D stuff. This is something called Area 15, which houses an archaeological site. Galt site in Central Texas. Researchers have unearthed projectile point technology never previously seen in North America, which they date to between 16,000 and 20,000 years old. In other words, a really long time between the 11 to 13-ish thousand years of the Clovis culture, which was once the oldest Native American find in North America. The story goes on. While clear evidence for the timing of the peopling of the Americas remains elusive, and I point out that's what it says here, elusive. Although there's pretty distinct genetic disconnect between Native Americans and the Asian continent 25,000 years ago. There's a growing number of pre-Clovis sites in South America, evidence of peoples in the Yukon over 20,000 years ago, a known glacier that covered much of North America in between, so elusive maybe, but also pretty clear. 2002, Area 15 of the Galt site in Central Texas was identified as an ideal area to search for remnants of early culture. This is actually got a Clovis site at it. The site features 5 distinct layers in the stratigraphic profile that showcase different cultural components. So, based on optically stimulated luminescence, I don't know what that is. Have you seen what is a optically stimulated luminescence dating system? I don't know this one.
I have not seen that, no.
Yeah. I wonder if this is the one that the Max Planck folks were using on the cave walls.
Oh, yeah. Well, yeah. I mean, they're going to optically stimulated. That means that they're shining light on it, but it's going to have a certain amount of fluorescence itself. And so that light triggering the release of light probably they're probably doing black lights, using black light. I don't know.
The sediment samples, approximately 16 to 20,000 years old. They discovered ancient material at the Loess deposit, including small projectile point technology. There was biface stone tools, blade and core tools, and flake tools. So they compared these Galt artifacts to Clovis tools. They found that the blade and core tradition are similar to Clovis blade and cores, which means that they were likely kept making those until the time of Clovis. But the biface traditions underwent some changes, and some of the early projectile point technology, they say, is unrelated, or at least disappeared, whether it became forgotten technology or outmoded in some way, is unrelated to Clovis. So, yeah, they just keep on finding people were everywhere that we thought people weren't. Isn't it at some point we're going to figure out that like people or hominins were everywhere in the world for not just 20,000 years, millions of years, the whole planet's been full of people. We keep thinking, oh, we discovered the thing. And no Native
full.
Americans were there on that continent
They
for
were-
20,000 years. It's not. Oh, but then we're going to figure out that there was some other, Homo erectus habilis ancestor at some point. I'm sure we'll find one in North America or South America. And it's like, oh, gosh, look, there were people there 3 million years ago. And they just have been people everywhere throughout all of time.
Yeah. So the optically stimulated luminescence does measure doses from ionizing radiation. It's used to date minerals as opposed to organic materials, which radiocarbon dating is used for. And it gives accurate readings for X-ray and gamma ray photons with energies ranging from 5 kiloelectron volts to greater than 40 mega electron volts. Yeah, so it looks at these radiative X-rays and gamma rays and makes use of electrons trapped between the valence and conduction bands in the crystalline structure of certain minerals, most commonly quartz and feldspar. Now you know.
Now I do.
And now
I need one.
I'd like one of those. Yes, please.
I'll take 2.
People everywhere. Yeah, and living up here in the Pacific Northwest, it is very striking. And I'm sure even as you get further north, even than Oregon, Washington, it is very striking the history. You go to natural history museums and the artifacts and the dating and timing of, what we know about the people that populated this area and how long they were here before, the 1800s when the white man came over and said, "This is ours now. Clear off." There was a lot going on before the last couple of hundred years.
Mhm.
Absolutely. Speaking of a lot going on, not just here in the Pacific Northwest, but all over America, we've got dogs. Dogs, supposed to be man's best friend, right? This idea that dogs, American dogs, anyway, there's this idea that they descended from wolves. And anyone who's looked at a Siberian Husky or other dogs of that kind of wolf-like Arctic breeds, the Malamute, right? You look at these dogs and you go, oh, they're definitely descended from wolves for sure. However, a new study published this week in Science found that in the 15th century, when the Europeans were coming over and killing all the people who lived here natively with disease, well, that happened to their dogs too.
Oh.
Yeah, and so the pets that came with the Europeans replaced all of the dogs that were native to the Americas.
Wow. Oh, that's wild.
Yes. So I used to think American dogs, they were called pre-contact dogs, as in before European contact with the New World, and that they were domesticated from North American wolves. However, this new study suggests that that there was a effect of disease, cultural persecution, and biological changes. And the history of humans is mirrored in our domestic animals. Just as indigenous peoples in the Americas were displaced by European colonists, the same is true of their dogs.
I still like to picture large packs of chihuahuas.
Yep.
In the hundreds going after bison on the open plain.
Chihuahuas, the scourge.
They were sneaky too, because the tall grass of the open plains, you never saw them.
The herds of the roving chihuahua.
And they were all on your ankles.
That's right. This study, they did a genomic analysis of 9,000-year-old dog remains taken from excavations in Illinois and other areas in North America and Siberia, revealing that the dogs originated in Siberia. And these 9,000-year-old fossil dogs came with the New World Americans, the ancient humans, as they crossed from Siberia, the Bering Strait, into the Americas. But those dogs, none of the mitochondrial genes that they looked at exist in dogs of present day. So, dogs of the past, goodbye. You were replaced. Replaced. Poor doggies. Fascinating history. Fascinating. Tell me about horse snorts. Is it time for horse snorts?
Yeah, well, it's a pretty straightforward story. Horses produce more snorts when they're happy.
Oh, happy horses.
That's their horse's way of telling you everything's cool. I'm happy. So I guess they were saying that there has been anecdotal reports of this. Horses snorting in positive situations. But this one, they went and studied it. They wanted to say, okay, well, let's see if this is really true. And so they looked at horses in riding schools when they were out in the pasture versus when they were in stalls, and found when they were out there in the pastures, they had more snorts. They also had snorts when their ears were forward, which is the horse's I'm in a good mood ear pose. When their ears were back, or under stress, not so much snorting going on. So taking it all together, comparing different contexts of when horses snort, snorting is now can be considered a reliable indicator of a positive emotion in a horse.
Good to know. Next time I am hanging out with horses,
Yeah.
if I don't hear the snorting, your horse is not very happy. I'm sorry.
I heard your horse snort once.
A snort and a stomp, and off that happy horse went.
Actually, this has nothing to do with the story, except that the story was conducted by Mathilde Stomp of Université de Reims, France, in college. So it was Dr. Stomp doing
Dr.
the
Stomp.
snort study on horses.
Stomp and snort.
That just happened to be.
I didn't know that.
Yeah.
Well done. Well done. All right. Well, we got dogs and horses in there. We might as well say we had an animal
Blair's
corner.
Animal corner right there, right?
Just like it.
Yeah. Blair's Animal Corner with Kiki and Justin.
And not Blair.
Not Blair.
But not Blair this week. No. All right, everyone. We are going to take a very quick break, and we will be back in just a few moments with more This Week in Science.
Can't explain the things you've heard more than intuition. The line of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see. The answers lie somewhere within this scattered plot.
Hey, everybody. Thank you so much for listening or watching. If you're watching live right now, thank you so much for joining us for another episode of This Week in Science. Now, if you want to see This Week in Science continue and thrive, let's talk about support. You are our support. You, our listeners, are the only way we bring any money in. We do not take advertising, and we love that. We love that we can continue to do this show, that all the things that we need to make this show happen are supported by you, that you like this show, that you love this show, that you want this show to be a part of your life enough to continue to support us. Now, you don't know how already. How can you support us? How can you help This Week in Science? Well, the first thing that you can do is go to TWIS.org, TWIS.org. That is our website, and it is where you can send people who need to subscribe to This Week in Science, who have not subscribed already. If you haven't subscribed, that is a great place to go. There is a big orange subscribe button. You click on it. It gives you options for the Google Play podcast portal, iTunes, and YouTube for you to subscribe and get TWIS in your device every single week as soon as we publish it. Now, additionally, at TWIS.org, you can access our Zazzle store. You need a new t-shirt? This is where you go. You can go get a TWIS t-shirt with our logo on the front, and it's a Zazzle store. So the t-shirt, I believe you can switch the color around, and you can put that logo on just about any color t-shirt of your choice. There are also other images from Blair's Animal Corner calendars from past years. There are mouse pads. There are luggage tags. There are lumbar pillows. We also have polo shirts, mugs, hats, tote bags, phone covers, all sorts of items with This Week in Science related logos and imagery, and a portion of the proceeds comes back to support TWIS. So by purchasing a Zazzle item, you are really helping us keep this show going. Back at TWIS.org, you can also donate directly to our needs by clicking the donate button. The donate button will take you to a PayPal interface where you can choose an amount of support based on what you are able to give. Or you can click on any recent episode, scroll down through the show notes. And did you know that we have show notes for every show? The disclaimers in there, all of the links to the stories are all in there. And if you scroll all the way through, there are pink buttons, again, leading you to PayPal interfaces to set up recurring PayPal donations so that you can have a set amount be taken from your account and transferred to our PayPal account every month. So you can set it and forget it if that's the way you want to roll. Makes it easy. So you don't have to, you can do it once or you can do it multiple times. Back at the main page for TWIS.org, you can also, or just in the header bar, click on the Patreon link. Patreon is a crowdfunding website that we use where we get where we're able to interact with you in an ongoing basis. I can send you little notes. There are I send little videos and little notes to everyone on occasion, and you can choose your level of support. You are charged once a month, so you can start at $1 a month. You can go all the way up to $1,000 a month if you are so able, and each level of support comes with its own perks. So whatever you are able to give, whatever level at which you are able to help TWIS meet our needs to continue, we do appreciate it. Also, over on, I'm going to put the YouTube channel up there. I see you YouTube chatters. If you're over on YouTube, you can also donate through YouTube. You can give us a little tip in our tip jar through the YouTube interface. So there are many ways to help TWIS. We appreciate you being here no matter what. Your help really does allow us to do everything that we do. And we really, really could not do any of this without you. Thank you so much for your support.
When they hired me to work.
Whoop, I got the wrong song. That's not a
How was
song.
that?
That's an old TWIS episode.
What was that? Sounded like some crusty old kook.
Yeah, who was that?
Can't explain the things you've heard more than intuition. A lot of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see. The answers lie somewhere within this scattered plot of words. Consumption word correct, disprove the rest. It's hot. The methods of hypothesis and patience are the only things I need.
And we are back with more This Week in Science.
Yes, we are. I'm so glad we are back here, and we are continuing to bring you more science to finish out this show. But right now it's time for What Has Science Done For Me Lately? This letter is from Logan Fette, and Logan writes in to say, "Hello, Dr. Kiki, Blair, and Justin. Science has been doing a lot for me lately. Multiple sclerosis has been prevalent in my family. I recently had gone to see a neurologist at the Mayo Clinic, and they checked to see if I indeed have multiple sclerosis myself because I had been exhibiting a fair amount of symptoms. I'm still awaiting the test results, but I know that science has made progress in the understanding of multiple sclerosis, and it's not quite as scary as it once had been. So even if I do have it, I will be okay because I believe in science allowing me to live a normal life, nevertheless, with the help of immunosuppressive therapies and so on." Logan, thank you so much for writing in with your story. And I think that is something that is so true for so many diseases where once there were no treatments, where we didn't understand what was going on. There might not be a cure, but science is figuring out ways to make your life better and other people like you. And so I don't know if you've gotten those test results back, but I'm wishing you the best in all of that. And thank you so much for sharing.
But if you're going to get diagnosed with a disease, now is always the best time versus some time in the past. Any time in the past was worse than right now. Right now is the best time. I don't care what the diagnosis or how dire it may seem. You're probably operating under an old assumption about the treatments of anything when we hear these things, right? We hear these scary diagnoses, and we think that means just horrible and nothing can be done, which was true probably when you first heard about this disease, right? But thankfully, science is doing a lot for all of us every day.
Every day. And all of you out there, thank you so much for sharing your stories over the last year and continuing to share your stories. We are still doing this, and I love getting to share with all of you. So keep it up. Keep sending me your stories. You can leave me a message on our Facebook page. That's facebook.com/thisweekinscience. Just leave a message there. Or you can email me at Kirsten, KIRSTEN, at thisweekinscience.com. We're going to keep filling this segment of the show with stories from you. So keep sending them, please. I want more. All right, 15 minutes to a better brain. How is science going to make my brain smarter, better, faster?
Is that what's next? Okay.
Or do you want me to tell a story?
No.
I'm ready. 15 minutes. Let's do
My
it.
rundown is frozen. I don't know why that is. This is a recent study in neuroimage demonstrating that exercise performed immediately after practicing a new motor skill improves its long-term retention. As little as a single 15-minute bout of cardiovascular exercise after you do the new task increased your brain connectivity and efficiency in remembering it. So this is Mark Roig in his earlier work. He's the senior author of the study. He demonstrated that exercise helps consolidate muscle and motor memory. So then he and a McGill-based research team decided to figure out why that was. We have these things called a reasonable or an interesting correlation that we find in science. We say these aren't science, but this is what science, this is like the footprints that Sherlock Holmes can then investigate. There's two footprints, not just one. That's a clue. Now we can better investigate, right? Now we can better drill down on what's going on. So they went to work trying to figure out why this is. They got some study participants together and they had them perform some different tasks. The first they called the pinch task. And this was grabbing a joystick, which they called a dynamometer, and they used it with various degrees of force to move a cursor on a screen, and they would put these rectangles together on the computer screen, and that was success. And I guess they could vary the amount of tension that was required on this joystick. And then they followed this by 15 minutes of either exercise or rest. They had participants off in these two different directions. Participants were then asked to repeat a new version known as the hand grip at intervals of 30, 60, and 90 minutes after exercise or rest. And this task, they would repeatedly grip the dynamometer for a few seconds with a similar degree of force that was used to reach the targets in the pinch test, just sort of to increase muscle memory, but they weren't repeating the task quite yet. They were just sort of kind of going through the motions of it. The final step of the study involved participants in both groups repeating the pinch task at 8 and then again at 24 hours after initially performing it. This allowed researchers to capture and compare brain activity and connectivity as the motor memories were being consolidated. So the researchers discovered that those who had exercised were consistently better able to repeat the pinch task, connecting different areas of the brain more efficiently, and actually exhibited less brain activity than those who hadn't exercised. Why did they have less brain activity? It seems like they've just perhaps reduced the amount of brain activity that correlated to it because the memory of how to do it was better retained. So they weren't trying to figure it out. They were just relying on now a knowledge. And that was true at 24 hours. What's interesting is that they weren't really any better or different from each other at the 8-hour test. So 8 hours after this, those that exercised, those that rested, no difference. It wasn't until the next day that there was a very clear marked change. So it looks like sleep is also important in sort of solidifying this new task memory. So it's 15 minutes of exercise after the new motor skill test, plus a good night's sleep.
Yeah, we've known for a while in terms of sleep that it's very important in the memory consolidation process, and that there are neural mechanisms that take place while you sleep to help solidify those memories, or what they're called consolidate, take them out of short-term memory, move them really into long-term memory. I think that's part of like what the switching between your deep sleep and the REM sleep might be, that this memory consolidation is occurring during these different phases of sleep. But the exercise aspect is really interesting. So the question is, what exactly this bout of exercise after you learn something, what does it do? We've been hearing stuff recently about the muscles in the legs helping you think better, being that there's a link between blood flow and exercise. So is it increased blood flow is stimulating the brain or helping the brain to do its job better, to consolidate more efficiently? What is going on?
So this is Fabian Damaso, who's Damaso, first author on the paper, says because of the neural activation in the brains of those who had exercised was much slower, the neural resources could then be put to other tasks. Exercise may help free up part of your brain to do other things. So that phrase right there, and maybe the increased blood flow too, reminds me of the study of the students who studied the material and then those who just went home and went to bed versus those who went and consumed alcoholic beverages. Because there wasn't more competing information throughout the rest of their day, right? It was sort of a turn-off time for the alcohol consumers. They actually retained the information that they had learned the day before better in that scenario. So, but now alcohol also, I think, doesn't it work a little bit as a blood thinner?
Yes, it does.
which might allow more blood to the brain. I don't know. That's the second though. But the first one is like maybe exercise is performing a similar function that alcohol performed in the other scenario, which is not competing with new memory to directly after the task, right? And so with no other things for that memory part of our brain to be focused on or working with, this is the last task you were given, learn this, and you keep on doing it throughout the night until there's something new to do. Right? One
so
possibility.
So the question then would be, I guess, does 15 minutes of meditation have the same effect as 15 minutes of exercise?
If you're a quiet meditator.
Right.
This is important. Some people's meditation, just speaking from my own personal, as soon as everything gets quiet, the mind starts racing, right? As soon as my body stops doing stuff, mind takes off. Blah. So I think it would, I think you could possibly apply this because one with the alcohol was studying just information versus this is a motor task. And I get that he's focused on motor task because that's how I initially came to some of his discovery. But I'm kind of curious then if the same thing couldn't be repeated with material read from a book or during a lecture. Like you get the lecture and some people rest and then some people go on a 15-minute run if you would see a similar result. My speculative tentacles are thinking yes, that you could see it in that scenario as well.
Could I spend 15 minutes eating spaghetti?
I don't
have the same effect.
But no, this is now if you're a listener of TWIS, if you like the information you hear here, but wish that you retained more of it than you do,
go for a walk. Go for a
run. Go for a little 15-minute jog after every episode.
Out there.
Some jumping jacks. And maybe the show will be there.
This study though is specifically related to motor skill learning, motor memory.
But again,
so it's
I think that's
all information.
No, it isn't. But I think it's because this particular researcher has been working on muscle memory that this is why it's connected. I think it's going to end up being this is a function of the brain.
Possibly. Yeah. Okay, prediction made here today by Justin. But seriously, 15 minutes of exercise. Practice your piano and then go for a run.
Yeah, no, that's I think that's a great
combo.
Idea. Yeah.
Absolutely. All right, I've got a story about the stuff we breathe. Well, air, but very specifically a component of air, oxygen. The oxygen that we know to be very important to life on this planet.
Much life.
Yeah, most life on the planet. Exactly. Not anoxic species. But there was an event 2.4 billion years ago called the Great Oxidation Event, where that was basically when oxygen levels became consistent planet-wide.
Giant algal ocean party.
Yeah, algal ocean party. Oxygen levels increased around the globe, and we kind of target that as like the start of when complex life really could get a foothold and get going.
That was the terraforming of planet Earth for what we for the predominant life forms that we are aware of.
And so the question is, what happened before then, and there's this idea that, okay, water on our planet might have come here from comets. But where did all this oxygen come from? And just did it slowly increase and just suddenly there it was? Or was there some variation in how it got to that great oxidation event? And now there is new research out of the University of Washington that found that oxygen levels was like a teeter-totter. They went up and then they went down. They went up and they went down. They'd rise and fall multiple times for millions of years before the Great Oxidation Event occurred. So there was
probably toxic to whatever was like there might have been life on like land already, like microbial, what have you. maybe a few plants, but they didn't interact with oxygen well. And then suddenly oxygen's in the air, it's getting everywhere. If things are dying off, which stops producing oxygen. So
right. So maybe things died off and then the oxygen levels fell again. And so maybe there was this process of the jump starting of life, as those false starts, like when you're trying to jump start a car. Okay. Okay, try again. Can you get enough charge through that battery to make it all work? And so these researchers, they say the production and destruction of oxygen in the ocean and atmosphere over time was a war with no evidence of a clear winner until the Great Oxidation Event. This is Matt Kohler, who's a doctoral student at UW in the Earth and Space Sciences, and he authored this paper that's in the Proceedings of the National Academy of Sciences. These transient oxygenation events were battles in the war when the balance tipped more in favor of oxygenation. And so, co-author on the paper found in 2007 that there was a small signal of oxygen about 50 to 100 million years before the Great Oxidation Event. And they found this by drilling into the Mount McRae Shale in Western Australia and looking for trace metals that are dependent on oxygen accumulation. And now this publication confirms a second appearance of oxygen about 150 million years earlier than that, about 2.66 billion years ago, that lasted for less than 50 million years. They used nitrogen isotopes and selenium as proxies for oxygen. These, again, were reliant on oxygen levels to accumulate in the environment. And Kohler goes on to say, what we have in this paper is another detection at high resolution of a transient whiff of oxygen. It's just a whiff. Nitrogen isotopes tell a story about oxygenation of the surface ocean, which goes to your point about where this stuff was happening. This oxygenation spans hundreds of kilometers across a marine basin and lasts for somewhere less than 50 million years. They did a couple of drilled cores looking into sedimentary rocks in this area in Western Australia called the Girona Formation. They analyzed various layers in these rocks, and they say they see a stepwise change in these isotopes, and that increased and then went back to zero again. And it's just this sudden signal of oxygen that was up and then it wasn't. And they talk about it as kind of like this, the rusting of rocks, that the oxygen was there and the rocks on the land, through oxidative weathering, they rusted and then they got transported to sea. And they say one of the strongest atmospheric biosignatures is thought to be oxygen, but this study confirms that during a planet's transition to becoming permanently oxygenated, its surface environments may be oxic, that's containing oxygen, for intervals of only a few million years and then slip back into anoxia. And so in terms of looking for life on other planets, if we fail to detect oxygen in a planet's atmosphere, that doesn't mean that the planet is uninhabited or even that it lacks photosynthetic life, merely that it just hasn't built up enough oxygen sources to overwhelm the sinks for the oxygen for any longer than a short interval.
Yeah, just check back in 150 million years.
Exactly. Just wait. Just wait it out. Planetary laboratories. That's right. Yeah, but that's the idea, that maybe there is microbial life. Maybe there is photosynthetic life. Maybe there are these organisms. They're getting their foothold, and it's just this little battle between the levels of the things that the different populations of things need, the anoxic species versus the oxic species, non-photosynthetic versus photosynthetic and oxygen producing. Very, interesting idea. Up and down. It was not just a consistent thing.
Speaking of things that were not consistent, scientific consortium led by Dr. Eleanor Skerry, British Academy postdoctoral fellow at the University of Oxford and researcher at the Max Planck Institute for the Science of Human History, has found that human ancestors were scattered across Africa and largely kept apart by a combination of diverse habitats, shifting environmental boundaries, and sometimes were separated for thousands of years, and that this gave rise to a staggering diversity of human forms, whose mixing over time, different areas, locations, eventually became the species that we are now. Our particular brand of hominin originated in Africa, but how? Many had assumed, as we teased about in the first half of the show, that there was a early human ancestor population that originated as a single, relatively large population, but as a somewhat homogeneous group that then slightly or slowly eventually evolved into us. Indications are now that what ultimately became modern human was always a multicultural affair. In a paper published in Trends in Ecology and Evolution, the usual study of bones, stones, and genes is aided by new, more detailed reconstructions of Africa's climates, the habitats over the last 300,000 years. So, stone tools and other artifacts. This is the material culture earmarks. Artifacts have remarkably clustered distributions in space and time, says Dr. Eleanor Scary. While there is a continental-wide trend towards more sophisticated material culture, this modernization clearly doesn't originate in one region or occur at one time period. Human fossils tell a similar story. When looking at the morphology of human bones over the last 300,000 years, there's a complex mix of archaic and modern features in different places at different times, says Professor Chris Stringer, who's a co-author in the study. As with the material culture, we do see a continental-wide trend towards the modern human form, but different modern features appear in different places at different times. And some archaic features are actually present until remarkably recently, he says. So when we think of what is a modern trait, maybe it's a brain casing or a brow or a whatever it is, shoulder or rib cage structure, what they're finding is, oh yeah, the brow's over there on that side of the continent. You got the shoulders showing up way down south. Right? Like, it's not one group. This is very sort of different. And I think we've talked about this before. The all peoples who are out of African descent for, the last, what would it be, 20, 30, 40,000 years, are very closely related to each other genetically. Within Africa, there's actually quite a diverse genome. And so when they did the genetic data, checked the genetic data for the continent, it also kind of agreed with this eventual coming together as a people. It's difficult to reconcile the genetic patterns we see in living Africans and in the DNA extracted from the bones of Africans who lived over the last 10,000 years, with there being one ancestral human population, says Professor Mark Thomas, who is a geneticist and co-author in the study as well. We see indications of reduced connectivity very deep into the past. Some very old genetic lineages, these are ones that are still current today, and levels of overall diversity that a single population would struggle to maintain. To understand why human populations were so subdivided, how these divisions changed through time, researchers looked at the past climates and environments of Africa, and they could see that there were shifting habitable zones that would isolate people in different areas over different times. So there's some of the most inhospitable regions in Africa today, the Sahara Desert, which is actually where my ancestors were lumberjacks. We were really good. We were really good at lumberjacks. A little too good, some would say. But no, yeah, Sahara were once wet and green, with interwoven networks of lakes and rivers, abundant wildlife. Some tropical regions that are humid and green today were once arid. So you can sort of picture as the wet, green, arid, dry, shifting environments move around, people will move around. You also see this actually within animal communities and populations, that these subdivisions sort of drive species to have different phylogenetic patterns in their distribution. So it's not just our animal ancestors, it's other animal ancestors as well that went through this. Convergent evidence from these different fields stresses the importance of considering population structure or models of human evolution, says another co-author, Dr. Lunas Chiki. This complex history of population subdivision should thus lead us to question current models of ancient population size changes and perhaps reinterpret some of the old bottlenecks as not so much bottlenecks in population, but in changes in connectivity to other existing groups. The evolution of human populations in Africa was multi-regional. Our ancestry was multi-ethnic. The evolution of our material culture was as well multicultural. Says Dr. Scary, we need to look at all regions in Africa to understand human evolution.
Yeah, got to take that broad look. Yeah. Oh, I love it. The evidence, the ideas that people are putting together now, it's shifting the way that, we don't, the way that we look at our own evolution will shift the way that we look at ourselves today. And I think this is a good thing.
Yeah. I got to tell you though, and the first time an anthropologist friend had the book open and it showed the linear progression through morphologies of skulls and what have you.
Yeah.
I thought it was nonsense.
Like, really? It was a straight line? It just went from that one to
then one day
us?
Homo erectus they gave birth to, and then those genes took over in that population, and therefore we're all Homo erectus going over. It's like, no, there's a lot more people. They're going to be different, and if they can do it, they will, because they're people. People can get pretty unpicky, and today, when there's lots of options, way back when there weren't a lot of options, they probably weren't as picky. If it looked human enough, that was probably good enough, close enough. And
Close enough.
And so, yeah, like the hybridization, but also the isolation. And I love this. And I guess I never, I think that I was more, when it came to once modern humans sort of showed up, the single population, the bottlenecking is definitely something that I subscribe to and kind of believed in. Even though the rest of the evolution I could see as being braided, streamy stuff, a lot of the early population. But I love this whole idea that, different aspects that we consider uniquely modern human were dispersed across the continent. And now we know which ones came together to become us. But it didn't have to be this way. There's a whole lot of different types of versions with maybe idiosyncratically, but it didn't have to be this kind of human. It could have been any of the others. It still would have worked.
All the versions of us, yes. What about versions of planets?
How many you got?
Well, maybe 5 more. Yeah. Researchers publishing in Nature Astronomy looking at the asteroid belt have determined that there could have been 5 more planets in our solar system at one point in time. That the asteroid belt is made up of shards of early planets in our solar system. Yeah. So, the question is the asteroid belt a remnant of the dust cloud of, those early collisions between the planetesimals? What exactly is it? Is it this remnant of the early dust cloud that was our solar system that we formed from, or is it remnants of collisions? Which one, right? So the researchers looked at, they were predicting collisions with Earth from the asteroids that we know of in the asteroid belt, and they wanted to clarify what might be sending them on the trajectories that they are on. And they looked at approximately a bunch of these asteroids and found that approximately 500,000 components of the asteroid belt are slanted in shape. And because of their shape, that means their composition is probably different than what was originally thought. Stanley Dermott, who's involved in the study, said we couldn't think of any forces acting to produce that distribution. If a big asteroid is smashed up and has a high inclination, then those fragments have the same inclination. And so they have concluded that the remains of the asteroid within the asteroid belt used to be planets, planetesimals. And there might have been, based on the quantity of asteroids in the asteroid belt, that there might have been about 5 planets. Fascinating, right?
So, yeah, note to self when designing a solar system, set the orbits a little further apart so to avoid collisions. This is it. You know, some of your planets are going to take slightly different orbital paths. Not going to be perfectly round. Give them some clearance. So you don't have this happen to you.
Exactly. I mean, what happened is these planets probably tried to take hold and they started forming, but you've got the different gas giants with their massive gravitational fields, right? And you have Mars and Jupiter kind of on the opposite sides of the asteroid belt, but maybe there was a gas giant that was closer into the sun that moved out at some point, or maybe there was a bit of movement between the planets that ripped apart whatever planetesimals were in that area between what is now Mars and Jupiter and is now the asteroid belt. These galactic, these solar system level forces just, sent things like billiard balls smashing into each other.
Yeah, Jupiter's probably ate a few of them.
Yeah, Jupiter probably did. But still, this leaves the question of, we still need to figure out which of them potentially will cross paths with the Earth and when, and how big are they going to be? Yeah.
What's sort of interesting, though, is there does seem to be a pretty decent ratio. Like I don't know which one of the planets it was. I forgot now, but one of the outside of the 3, maybe it was the 4th, 5th, 6th, 7th, where it was out there. I don't remember. But one of the planet's positions was predicted within 10% before it was found in terms of its orbit.
Right.
And interestingly, when we found a lot of these exoplanets, our solar system with 8, 9, 10, however many you want to count or not count, is pretty average. That this is like, around 8 to 10 is kind of where most stars land with the number of planets going around them. And it probably does have to do with orbits, right? Like, just to have a healthy orbit, a healthy cleared orbit, and not run into something in the last 4 billion years, 5 billion, 10 billion, 20 billion, whatever. You got to have some space to breathe. And if you formed in the wrong spot, you combined, or you were turned into an asteroid field. So we'll likely see a pattern of our solar system repeated around many, many, many stars, which is actually what we've sort of been seeing.
And that's what we want to kind of look for, right? If it happened here, let's look for it other places, and maybe we'll find that rocky planet with water in the right place around its star. Yeah. How are we doing for stories? Do we have more stories?
I was all of the stories which I have brought.
I have one more quick story. There is research published in The Lancet. It was published July 7th, showing that an experimental HIV-1 vaccine regimen is well tolerated and generated comparable and robust immune responses against HIV in healthy adults and also in rhesus monkeys. And it protected against infection with an HIV-like virus in monkeys. This was a phase 1-ish clinical trial, basically, which is only to look at safety, not so much to efficacy. That's a phase 2 trial The phase 2 trial has been initiated in Southern Africa to determine safety and efficacy of this vaccine candidate in 2,600 women at risk for acquiring HIV. And this is one of only 5 experimental vaccine concepts that have progressed to efficacy trials in humans in the 35 years of the global HIV/AIDS epidemic. So this is big news that we have something progressing past phase 1, entering phase 2, and that there is a promise. In 35 years of the HIV epidemic, only 4 HIV vaccine concepts have been tested in humans. Only one provided evidence of protection in an efficacy trial, but it had such a low rate of protection, it wasn't really considered effective enough to advance the vaccine to common use. So still to be seen, but it's exciting to note that a new HIV vaccine is in the works. This could be big news globally as the HIV epidemic continues, even though we don't hear about it so much in the news anymore.
Well, isn't that though also part of like we were talking about from the What Has Science Done For Me Lately, that it's no longer really a death sentence.
Yeah, people live with it much better, especially in first world countries. But in third world countries, in the countries that don't have great medical care, it is a huge problem, and it is growing.
Which is why I haven't been hearing about it.
Yes, yeah. It's all the what news is coming out of where, when. Yes, but this is a vaccine candidate, and fingers crossed. So we will hear more in the next few years as this vaccine crosses all the barriers and hopefully gets put into use widely for people. Fingers crossed. But that does it for This Week in Science. So many amazing stories. Thank you, everyone, for joining us. Justin, thanks for a great show.
Thank you for having me on your great show.
You're welcome. So glad.
Both be on the show.
You taught me a lot about humanity today on the show. It's very kind of you. I would love to say thank you to all of you who have been watching us on the live stream. Thank you to those of you who are in our chat room. Hey, chat room, how's it going there? Hey to the YouTube crowd. Hello. I'm glad you were here chatting with us as well and with each other. And thanks for keeping it nice. We do appreciate that the chat is a nice place for people to come and share their time. Fada, thank you so much for your help with show notes, with social media, and with keeping our chat rooms happy and clean. To Identity for recording the show, and to Brandon for helping us out in simulcasting to Facebook. Everyone out there, I would also like to take this moment to say thank you to our Patreon sponsors. Thank you to Paul Disney, G. Burton Lattimore, Richard Onimus, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Andy Gross, Steve DeBell, Alex Wilson, Tony Steele, Ed Dyer, Craig Landon, John Ratnaswamy, Mark Mazzaros, Jack, Matthew Litwin, Jason Roberts, Bill K, Bob Calder, Time Jumper 319, Kyle Washington, Eric Knapp, Richard, Brian Condren, Aiden Jeff, Kevin Reardon, Dave Neighbor, Christoph Zuknurek, Noah Zemke, Jim Seabright, Ashish Pence, Jacqueline Boyster, Ulysses Adkins, Sarah Chavis, Richard Porter, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Slazewski, Jim DePoe, Greg Riley, Sean Lamb, Ben Rothig, Steve Lesseman, Kirk Larson, Robert Aston, Rudy Garcia, Marjorie, Gary S., Robert, Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, E.O., Mark, Tyrone Fong, and Keith Corsell. Thank you for all of your support on Patreon. And if any of you out there are interested in helping us out on Patreon, you can click the link at twist.org or go directly to patreon.com/thisweekinscience for more information. Remember that you can help us out simply by telling your friends about TWIS. On next week's show, we will be back at 8 p.m. Pacific time on Wednesday, broadcasting live online at twis.org/live. You can watch there and join our chat room. Don't worry, though, if you can't make it, because the show is preserved for posterity in many different forms. And you can always find us at twis.org/YouTube or just twis.org.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have one of those mobile type devices, you can look for TWIS, the number 4 Droid app in the Android marketplace, or simply This Week in Science in anything Apple marketplace.
And for more information on anything that you have heard here today, show notes are going to be available on our website. That's twis.org, where you can also make comments there and start conversation with the hosts and other listeners.
Or you can hit us up directly. Email Kirsten at thisweekinscience.com, Justin at twisminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jackson Fly, although that's mostly politics lately, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
And we will be back here next week. We hope that you will join us once again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you learned anything from the words that we said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in science. This week in science, science, science. This week in science. This week in science, science, science. This week in 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 science.
And now is time for the after show. Justin has left me yet again. Who's still at YouTube? Hello, YouTube. Who's there? Sunny de Oliveira, JD Goodwin. Yes, you just got here. Party's not quite over yet, but we did finish our faux show. Hello, chat room. Can I make that? No, it's not going to switch over to the chat room. I have to do that separately. But I can stop this. Stop this. Stop this madness. Hey, everybody. Welcome back. I hope everyone had a wonderful 4th of July. If you're in the United States of America, and maybe if you're in other places that you had other nice holidays or even just a nice weekend. I hope that you're excited to see This Week in Science back again after a week break. We're back. TWIS monkeys. I need I have my monkey shirt on. I like my monkey shirt. I'm watching you. That's right, Hot Rod. I see you in the chat room. I know what you did in the chat room. I saw it. I do. I watch it. I watch it all. I try to see everything. The all-seeing Kiki. So I had a fun experience this last week. I got to, yay, it was nice. Good, Fada. And Fada, thank you for all those awesome social media posts doing retro TWIS stuff, bringing people back to other older interviews and old disclaimers and a day of what has science done for me lately posts. Oh my gosh, that was wonderful. I think we need to do more of that. But just during that week, that was awesome. It was fantastic. I like that themed up social media activity. It was fantastic.
I'm glad we overcame some technical issues at the beginning there, too.
I know, I am too. I'm so glad that we were able to make it work.
Momentarily terrified that I was.
I'm like, oh, what is wrong with the internet? Yeah, just turn it off, turn it back on, wiggle it a little. Fine. Yeah, it was good. It was good. Should I do a speed test while we're sitting here? What's my speed, Casey Jones?
Trouble ahead.
Trouble behind. And you know that notion
just crossed my mind.
Yeah. 58, 59. 59. I should have more than that. I should have more than that. It's enough for this, but not enough for what I want to do. I need more. I've got to have more power. Yeah. Upload 11.19, download 59.92, ping 10 milliseconds.
Is that good? I would not know.
It's okay. It's okay. But when you want to be streaming live, you want more fasterness.
Always.
So I had a fun experience on Monday. Got to do another shoot day for a TV show called Strange Evidence.
Nice.
Yeah. And it's a show where they take videos, like security cam footage, people's cell phones, things basically that have been uploaded to the internet, but that show strange things happening that are inexplicable. Yeah, so I had fun. I got to talk about things like the skunk ape.
Did like Photoshop come up a lot?
We didn't, not too much actually. There were a couple of times where I was like, yeah, I think that's probably been doctored, that maybe they used modern animation and graphic technology to be able to add that in there. That maybe
Mhm.
this was done using special effects, maybe.
Mhm.
Yeah. But it was fun. It was fun. You know, there were lots of clips of me saying things like, some people say.
Yeah, that's kind of a throwaway, meaningless word. Some people say.
Some people
That's dangerous.
say.
That's dangerous territory you wandered into there, Kiki.
Oh, I did, and it's so great. I have no qualms about it. I don't even care. I think it's fun television, and it was funny.
Some people say I
Fun.
am the greatest broadcaster in the history of podcasting.
Right, there we go.
As some people may be me and my kids, but they are some people who say this.
There are some people in this world. Some people got opinions. Hello, after show. Hello, Justin Steiner. Welcome. Carrie Rutens. Who else is in there? Old Scratch. That's an awesome name. All of our chat roomers. Gord, Identity, and Chat, Beef, CR1, Dale Poco, Dave Shorty, Ed from Connecticut, Electro 33, Epony, Fada, Flying Out, Growly Bear, Hot Rod, Huffer, Janiscu, Kevin, Unique, Mav HQ, Noodles, Oleum, Protonic, Cordina Mouse, Rorks, Paradox, or Paradox, Strengths, all you people. Woot! Thanks for being in here. Thank you for being in here in this moment and letting us know. You know, if it weren't for you, we wouldn't have known about our buffering issues.
Yes.
And if we were just recording a show and putting it out there, if it weren't for you, there would have been nothing for the podcast later. Because Justin and I were having absolutely no issues.
Yep.
Yeah, it was just the external feed. It was Google. It wasn't us. It must have been. Or it was one of us. I have no idea. No idea.
Oh, shoot.
What? What shoot?
Oh. So I'm doing a speed test, but I also realized like I'm live streaming right now while doing the speed test. So is it really fair?
I don't think that matters, does it?
I think that should be what matters. My ping is 10. Is that a good ping? I don't know what a good ping is.
That's my ping.
Okay, I got the same ping. My down ridiculous. It's 67 plus Mbps's, which I'm thinking is megabytes per second.
Mm-hmm.
My up 8.61.
That's good too. You're good.
Is it good? So I'm good to go. Okay.
Fine. It's fine. It's fine.
I've got the speedy internet going here.
Speedy internet. It's speedy internet. Ooh, tomorrow's going to be hot. How hot is it in Davis?
I have no idea, and I love not knowing.
Are you just indoors all the time in the air conditioning? You don't go outside?
In fact, I spent a decent portion of my day at 4C.
4C? That's freezing.
It was cold. It was a cold day. Yeah.
Oh my gosh.
It was chilly.
That's extremely chilly. You just do your work in the freezer?
I have. It's not quite the freezer, right? It's like I think it's what is that?
4C is cold.
Is it that cold?
Yeah, that's like okay. I don't do conversions very well.
Just get a little bit shivery in there. It's enough to do that.
Okay, so it's 39 degrees,
Yeah.
which is just above freezing.
Yeah.
Just above freezing. So it's not quite a freezer.
Yeah, no, not
Still.
quite. But it's yeah, it was.
It's not warm by any
but
means.
but that's how I like outdoors. You know, the sun was out, and I didn't see anybody jogging, so maybe it was hot. But wouldn't know. I spent decent part of the day. A little shiver on.
I think yeah. Has it hit the hundreds in Davis yet?
I don't
You have no
know.
idea.
This is what's amazing. I really don't know. And there was a day that I had off that got 200 maybe, right about there. Maybe it was just a little under. And I did go swimming. So
Swimming is
but I
nice.
waited till, I guess it was been the hottest part of the day because I waited till the afternoon.
Mm-hmm.
Wasn't that bad.
I want a pool. That would be nice. To find a pool.
Yeah, find one. Don't own one.
I don't want to own one. I want to find one.
Yes.
Yeah.
But you're in Portland. Like,
nobody has pools in Portland
why would
because it's
anybody
like
have a pool in Portland?
Why would you? I mean, it's like warm for a month.
Yeah, hot tubs, I imagine, are a big hit.
Yeah. Hot tubs all over the place. Pools? No. It's like a waste of space to have a pool in Portland. Ed from Connecticut wants to know if you're doing any interesting experiments at work.
Yes.
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 But you can't talk about them.
Cannot talk about them at all. Although one of the cool things that I'm getting to play with is the next generation sequencers
Ooh.
from Illumina. I can talk about that I'm using that equipment, which is kind of funny because now I've got a it's sort of like playing along at home for a lot of these are the same sequencers that Svante Paubo has used.
Yeah.
And sort of decoding the Neanderthal genome.
Yeah.
So I get to do a little of playing along at home, home, whatever it is, with getting to run those. But
that's cool.
And I had the experiment I was working on today is one that it's an experiment that's under construction. It's a designing a new experiment. So has to do with things that need to stay cold so that they do not perish or exhibit untoward activities. So yeah, so a lot of the setup and a little bit of monitoring took place in a room that is typically used for storage. But I converted it into an active laboratory today because that was the only way I could figure out how to keep everything at the temperature it needed to be while manipulating stuff.
I love it.
Yeah. So next time I will bring a sweater. I didn't have I didn't realize going in, I thought I'd just be able to just go in, set up, and then I realized it's just easier to do everything right here. And if I thought ahead, I would have been wearing thermals.
Right? Yeah. Instead of freezing yourself to death. That's how it is. That's how it is.
Oh, yes, Genescu. I can't wait to start sequencing Martians. That will be awesome.
Gotta get some return missions, right? Get some samples back from Mars. But do you think, okay, so, there was a movie, it came out last year that was about, a sample that had been had come from Mars or something like that, or from the moon. I don't remember exactly where. But there was a sample and they had it on the space station. And they're like, oh, let's observe it and let's do and they put it in all these different experiments and started doing stuff, and all of a sudden it started dividing and it started showing activity and it started growing, and of course it's an alien monster that wants to eat everything. And so that's the horror story aspect of it. But was it called Life, Sunny? Yeah, I think it might have been called Life. Yeah. And, there's the idea though is that they had all these fail safes to keep
So
that
I
sample from ever
just transitioned between thinking this was a real story and that I somehow missed.
A movie.
I missed the movie. I missed the little disclaimer in there that this was a piece of fiction. And I'm like, I should have heard about this. Of all the people, not only should I have heard about this, somebody should have asked me if I knew anything, minor details on it. Like, how did this not come up? Okay. All right. I get it. Now we're talking about a movie. Okay.
We're talking about a movie. But the aspect of it I thought was, more realistic. And then they broke it down and it was like they had to break it so that the alien would end up potentially making it to Earth, But the whole thing was this sample from someplace else in space. Do you actually bring it back to Earth? No, you don't. You keep it in space and you do all of the experiments in space. And that sample, there should never be a sample return mission to Earth if it has a potential of organic content, right?
Always bring it to Earth. It's fine.
Always.
Look.
No, you didn't see the alien. It was and it was eating.
Yeah, we have things that eat here. We've been eating things. If this thing's out in space, right? Wherever it came from, they failed. Right?
It ate everybody in space too.
That's why there's nobody in space.
That's why there's nobody
got eaten by this thing. I'd be more afraid of it getting devoured or consumed. I mean, you find some outlier organism, even if you go that far in space, there's something here that's probably got a predilection to eating it or devouring it or breaking it down.
Probably there would be something
more than the other way around.
The one thing, so not the one thing, but I thought it was really funny. My friends know I'm not a big horror movie fan. I'm not into the slash and gore and that kind of stuff. I like thrillers and some, a little bit of a edge of my seat kind of stuff.
Right. But I too don't want to watch the Surgery Channel.
So they're like, nicely trying to pick out a movie that I might want to watch. And they're like, oh, Kiki, it's a space movie, Life. Maybe, it's supposed to be a little scary. Do you think you can handle it? And I'm like, oh, maybe, and I'm expecting this terrifying, oh, so scary alien kind of movie. But I couldn't because I read about science all the time.
Right.
I got so annoyed with all the things that were wrong with their laboratory protocols and everything that I was so mad at so many things in the movie. I'm like, that's not the way that works. that wouldn't happen like that. That I couldn't get afraid of the movie because it destroyed my suspension of disbelief. Yeah. So sometimes these are the problems with being a scientist or a science enthusiast. When you try to enjoy a movie, it can be a problem.
Yeah. Like there's a lot of throwaway of this thing was genetically altered and therefore now it's 50 feet tall and wants to, swat helicopters out of the sky.
Right.
Nah. Really? Is that really that quickly? You just went from this to that? Okay. All right.
Yeah, I love the idea of the Mystery Science Theater 2000, 3000 type stuff, the commentary on movies. Oh my God, you sit next to me when I'm watching a movie that has anything to do with science. I don't shut up. I'm like, did you know that? I can't believe that. Yeah. Yeah, I need to watch those movies alone. Talk to myself. Ed says the expanse shows that anything we find in space, we will try to kill or we'll try and turn it into a weapon. Yeah.
Yeah, sounds about right.
Sounds about right. Yep. Pretty much. Hot Rod. Well, I saw a video of former missile silo operators telling stories of UFOs disabling missiles, and they were told to keep quiet for years. Makes you wonder.
Well, yeah, you
No,
don't
it
want to
doesn't.
get shot by a missile just for, driving by the planet. Be annoying.
Yeah. Ooh, so I'm wondering about the big conspiracy things right now. So the crazy sound weapon.
Oh yeah,
The sound weapon that was in that was potentially used in Cuba and then also in China. Right?
Oh, I didn't know that. I didn't hear that.
Yeah. So more recently, people at the American Embassy in China reported symptoms similar to what were reported in Cuba. So like kind of neurological problems and so there's this thought that there's some kind of sound weapon that's being used against Americans in other countries.
So
Dun,
that's really scary. And I was recently looking at an acoustic mixer. Right? So no impeller, no physical vibrating thingy, but just using sound to mix and agitate a material. And it's fast. There's a video on there somewhere. It's the RAM, which is something. Name of the company starts with an R. Acoustic mixer, and they put in this corn syrup and food coloring, and in 4 seconds, it's blended. It's so intense, and
that's cool.
Yeah, like I want 3 of these, right? But the thought crossed my mind, like, wow, this could potentially be weaponizable. In fact,
dangerous.
It was developed. It's a mixer for laboratories and stuff now, but and I guess it has a big following in lots of pharma and that weird form of pharma, which is I can't remember what it's called. Combinering. I don't know what they are. They do combinering of different compounds or different pharmaceuticals. But it was developed by the Department of Defense.
Interesting.
Yeah, that's it. What is this? This is
so RAM, the resonant acoustic mixer.
Acrylic with carbon black. So they use it a lot for coating things.
Interesting.
Yeah, but there's a video out there where it's just seconds.
And everything's evenly coated.
Yeah. Fascinating. There's one where they're doing the mixing. It's just intense.
Fascinating.
Yeah, I know. You get lots of like audio mixers when you try to find these things.
But it's resonant acoustic mixer. This is an advertisement, probably.
You had the right one there, though.
Here's some guy giving a presentation. What are you doing? You're talking. Talk, everybody. Evolution of mixing. What else is going on here? Oh, look at our mixer. We're going to put some stuff in it.
Eventually. Yeah, this isn't the one I want. I want the other one. The first one you had up there.
No, it looks like it. Look.
It kind of does.
They're going to put he's got just some white beads in there. And look, we're mixing this stuff up. I'm going to hit play and it's going to there, look at that. And then it's so vibraty, you can't even take video of it.
Right.
You can't even take a video of it because it vibrates so incredibly.
See if you can find the corn syrup one, because it's 4 seconds, but then they also do a slow motion video of it, and it's pretty wild.
They're mixing something else there. Corn syrup. What it was? Ram corn. That's mixed. Look, we made a mixture. Okay, I'll find a corn syrup thingy. But that's fascinating. You know, and this is ultrasound, right? Or
I don't know how to do it.
Resonant
is usually performed by something that's not diaphragm, like a speaker.
Shaking. Residine mixers. Oh, it's a PDF. That's not a video.
But that's Resident. That's the right place.
Yeah.
Look for videos. Like, click under videos.
Residine.
No, not these. None of these.
No.
You gotta put the Residine or the RAM in there.
Ooh, but look, let's look at toroidal vortices produced by 5, 4, 3, 2, skipping ad. Skip ad.
Oh, these are always fun.
Toroidal vortices created by dolphins, beluga whales, humpback whales. Is that what? Okay, there we go.
Yeah.
Those are interesting. Play with the air in the water, like creating
That's
different
awesome.
rings. It's so cool. Ow. Chomp, chomp. That's so neat though, but it's not, I mean, it's air, it's bubbles in the water, and they just know how to control these things. I don't know. Stop screen sharing. Stop screen sharing, watching the videos online.
So that's I don't know if that's a conspiracy thing. I think that could be a real thing, because I think that type of weapon is possible and
Totally, right?
frightening forensically, because it's not going to leave physical evidence. I mean, like, that you can easily backtrack to a source of, like, a ballistic or a chemical signal.
And there are sonic weapons that are in use that focus sound that the police use, right?
Crowd control.
For crowd control. Yeah.
V8.
That's fast. There's something going on there, and we don't know what. But I'm sure I'm guessing you could find so many conspiracy theories online. So the other one, what was the other thing that's going on? Oh yeah, in England, there had been a chemical attack in this one area of England, and recently there's just some random couple who got sick, and they think that there's either been another chemical attack or there's some kind of somebody left some bad chemicals just at a bus stop.
Well, right. The first was an assassination of a former Russian spy. And this is a neurotoxin that was developed by the Russians. So seems like that's a pretty easy connection to make, because, again, now we have a chemical trace there. So this other couple lives within a few miles of where that event took place. So yeah, somebody maybe discarded a a handkerchief at a park or left a residue of this chemical, right? And then
Right, but the
they've
thing is,
somehow,
it's been a couple of months though, so it
right?
shouldn't persist. Whatever it is shouldn't persist in the environment that long. It should have broken
It's
down.
right. Maybe it's on a door handle. Maybe it's,
Right?
maybe they hid it in a book and then ditched the
Something.
book, or
Who
like
are the question
something.
is, who are these people really?
Yeah, maybe it was them, right? Yeah, but like they don't. From what I understand, I haven't been following the story, but from what I initially got was that they don't seem to be people who would be targeted by the Russian government.
Exactly. They don't seem to be.
But it
Yeah, I'm just trying
makes
to
it.
spark more conspiracies here.
Well,
That's all.
okay, so here's the thing: get rid of the conspiracy stuff. This is not a coincidence.
No.
Right? This is beyond the pale of a coincidence that this, illegal has to be manufactured, is identified as being manufactured from a location used against the people connected to that location, and then it pops up again in the same area. Yeah, it could be a secondary contact of some sort. I would guess.
Yeah. Right? Yeah, it's got to be something like that. But I find it fascinating these these places where modern science kind of overlap into this world uh of intrigue and global espionage and this spy world. What is going on? Sonic weapons, chemical weapons. What is the science behind all of this and how can we piece it together using science and forensic evidence?
Well, I think it's just further evidence that the Cold War is back. We might have had a little hiatus, but the Cold War is back, and everybody seems to understand this except for one particular individual.
Yeah. What? What are you talking about? What? The World Cup is going to be not between England and France, as I had been hoping. I was hoping for some serious action there. But no, France and Croatia, which is going to be fantastic. Croatia.
What's a World Cup?
The soccer, football, goal. The World Cup. Big soccer competition between
Like a
various
fighting match?
countries around the world. The United
It's a
States
sports ball
doesn't
thing.
usually take
It's a
place.
sports ball related.
It is sports ball related. Yes.
Do they also have a portion of it where they sail on boats? Or is that a
No.
different cup?
There's many different sailing cups. Yeah. Right now, I think they just had the end of the Pacific Cup, which was a race across the Pacific Ocean from San Francisco to Hawaii. And now there are people celebrating having made it across the Pacific Ocean to Hawaii in their boats. I would like to go to Hawaii and celebrate with them. I think that would be nice. I don't think I want to sail there in a boat, though. Could go in a plane, something like that.
That's like 5 hours by plane, isn't it? That's got to be a heck of a
super easy, right?
Trip.
That's quite a sailing trip.
Isn't it?
Mm-hmm. Exactly. Thank you, Janiscu. FIFA. Yeah, FIFA worldwide. Right. And Dave Shorty, yes, it's an actual World Cup. Except, yeah, there's not many who don't take part in the World Cup. But I think the United States is one of those. We don't play that silly football game. You don't play with our pigskin, we won't play with yours.
That one's just called the Super Bowl. It's
It is
not.
just
They
the
don't
Super Bowl.
claim it to be the World Bowl.
No.
You're thinking of the baseball people. So that sports ball.
Well, World Series.
Just because I think Canada has a team.
Mm-hmm.
I think. I'm not even clear on that. I'm not even sure. My sports ball knowledge doesn't even go far enough to tell you whether or not Canada has a major sports ball team.
Canada does have a major sports ball team. It is true. I know that. It's true. Yeah, the US is into World Cup stuff, but the women's is not the same as the men's stuff. And no, US doesn't take part in the World Cup as far as I'm aware. We really haven't ever. Just not in it. Not in it. All this global
Soccer is a thing that
stuff.
all Americans, I think, are into until about the age 12. And then we stop for no apparent reason and nobody questions it. Like there are huge swaths of otherwise valuable, usable, healthy real estate throughout this town dedicated to the soccer football fields. And you
we all need our.
and you look.
You need your 15 minutes to remember things.
From like 6, 7, 8 to about 12-ish. There's kids out there playing. They call moms. The thing they call moms is soccer moms because they're taking kids to soccer all the time. But then at some point, again, and nobody questions this, we all just stop playing soccer. And we're done. And we don't watch it. We don't think about it. We don't carry on soccer playing, soccer ball watching, nothing. It's just over for Americans. That is kind of strange. Actually.
It's over. Yeah, it's true. It's true. Yeah, it is this big thing in childhood and then it just disappears.
Like there are 100 times more kids playing soccer than football. American.
Mm-hmm.
I would say 60 times more, 50 times more. I don't know. Maybe those numbers are too high, but a lot more than play baseball or softball. Lot more.
Yeah.
Soccer is like the national pastime of childhood athletics in the United States. But then we're done. We never look back.
Oh, yeah, and Ed, there's a plane flying awfully low overhead. Anyway, Ed from Connecticut, the Robo Games competition is indeed a world competition. Countries from all over the world come to California to compete. It is indeed a world Robo Games. Lots of cool. It's cool. It's cool. Mm-hmm. Yes, and soccer is it's very inexpensive sport as well. You just need a ball. It's pretty easy
and
to get
patience.
and patience and gotta practice. Yes. The kids trapped in the Thai cave were rescued without the use of Elon Musk's submarine. Did you hear about this, Justin?
Which part? I heard that Elon had a submarine he wanted to test on some children. I heard he went as far as trapping them into one of his underground compounds.
His lairs, really.
I don't think it's the same one where he's already mass produced the what is it, the 3 Tesla 3. Yeah, but yeah, this is so happy that this has got a happy ending to the story.
I know. I am like, this is some good news that the world needs. That's why everyone has been so focused on it, I think. But Yeah. Everybody is out of the cave safely. Hopefully their health is all good and that they all make it out onto onward and upward to play soccer some more.
Traumatized, claustrophobic.
Although I was thinking about this cave that these kids were in, they had to crawl through little tiny spaces, 15-inch wide channels, really, tunnels, to get to the point where they were trapped. So whose great idea was that? The soccer coach was like, "Yeah, let's go do that. Let's just keep climbing and going deeper into this cave."
Well,
I would be pretty annoyed if my child were on a soccer team trip and I found out that the coach allowed that kind of caving. Go to a cave where you go underground and it's big and it's open and it's wide, but don't go through it. What? You're going through a little tiny. Don't do that. No.
Yeah.
No.
Again, I don't usually follow this sort of stuff, but I did happen to hear something about the coach saying that he wasn't aware that it was going to be like it was. Like he thought they were going to go maybe walk into a cave and be like, "Oh, here's a cave." And then, once one or two of the kids slips through, you got to go after them. You got to keep the group together, right? And, I don't know. That's
Glenn P says they went deeper because the water was raising. I guess that would be a good reason to do that.
Yes.
But still, you don't know exactly where you're going. But oh man.
But I think is it possible they were partially saved by pumps? Basically, what is this? Like sump pumps and stuff, or inline pumps that were pumping water out.
Pumping water out of the caves, yes.
Yeah, Elon, make one of those, and then you'll be useful.
That's right.
Testing submarines on children. That is kind of I didn't get to see his who knew he developed a little personal submarine for kids? Like that's what else is he working on? He just hasn't bothered to come out with it. He has a child-sized submarine? I
think they designed it. They had the idea, maybe. I mean, with the boring company, they're digging holes underneath things and thinking about small spaces and caves and tunnels, and
so they might need the emergency extraction for flooded tunnels, that sort of thing.
Yeah.
Okay.
Yeah, and the idea is that it's a small device that could be controllable, you could hold on to it, and it would hold oxygen,
like in the
and
intro to
supplies
Johnny
that
Quest.
you need. Yeah, and then it would just pull you through spaces. Yeah. Child-sized, and they sent a small one, knowing that the tunnels were very small. Yeah, everybody's saying the kids also did it without the coach. Yeah. Children, why do you want to go put yourself in small places? Put yourself in small places. This is why I don't know if I will ever go caving again.
Yeah, it's not one of the things that appealed to me because usually if I'm going to start off on a destination, I get climbing a ridge line, right? Once I'm up there, I'm going to get treated to a view. If I'm going down into a cave, it's just going to get darker and cavier, right?
I tell you, we went to the lava caves, the lava tubes in outside of Bend, Oregon, as someone mentioned also in the comments, Matthew Litwin, right? I didn't do that in the lava tubes in Bend, Oregon. I didn't climb into the small places, but even so, just being in this lava tube underground, at any minute, even though this tube has been here for a long time, there could be an earthquake, there could be a cave-in, I could be trapped underground, under rock. I was very uncomfortable with the entire situation. And then I'm imagining, all these people, and this is the kind of cave that you can stand upright in. There's it's not a difficult cave to explore. I'm just thinking about crawling through these small places, about researchers and also videographers for science documentaries who who dive in these underwater caves, and they put themselves into these tiny spaces where they have to take their oxygen off their back and like put the tanks in front of them and then push them through as they swim through these spaces that are just barely big enough for themselves.
Always
And
remember the buddy system. Don't spelunk alone.
I know. Oh my gosh, I am amazed at the persistence and the
So my
I guess
uncle
is it courage? Is it
my uncle was a
courage?
miner,
Yeah.
and there was one job site in Utah. I believe it may have been a uranium mine, in fact. But he was explaining that it's a mile and a half, sort of to the work site. It's some of it's down, some of it's across. But they have like a mini railroad cart type thing that drives them there. But it takes a half an hour because it moves slow. It's not like you're on the freeway. It's a slow moving ride.
Yeah.
Just
waiting.
But it's a half an hour of descending, going deep into the. I think it was found it to be a little bit on the unnerving side. And he's a miner, so he's used to going into these places. His job was he called it being a powder monkey. So somebody would drill holes, and then he'd go and fill them with explosives of some black powder usually, and then blast out sections underground. So, but wouldn't have to, thankfully, be standing right next to it when it does that. He runs out a nice long fuse line or igniter line, and then would set it off. But that would be, no. Got a 2 mile an hour cart, whatever. If you've been a 3 mile an hour cart,
go faster. I'm giving her all I can, Captain. Oh my goodness. Oh my goodness.
Must be quiet down there though.
Yeah, probably so quiet and dark.
Yeah.
Fan of the dark. Yeah. Like sometimes I wonder about this idea that our ancestors spent a lot of time in caves. Yes, maybe they were safe places because you've got walls, you've got a little shelter, you're not getting rained on. I get that. But honestly, I mean, I don't know. I think enough people have like fears of caves. I mean, we talk about this like intrinsic fear of spiders, intrinsic fear of snakes, these poisonous things that have been with us forever and like things that we're concerned about. Also, why don't we talk about fear of caves, claustrophobia? Like, did that come from somewhere? Ancestors being killed in caves.
I don't think they were in the caves. I don't think they liked it then either. Think about that.
They liked it.
With the very few examples where there was no choice, humans avoided cave dwelling. That was more of a Neanderthal thing, right?
Right? Yeah.
We keep finding out the cave paintings that the early Homo sapiens, the modern. No, those are Neanderthal paintings. That wasn't even us. We avoided caves. But I guess it depends on if there's something scary outside of the cave. Something scarier than the cave.
Right? Yeah.
Because really,
dangerous things outside.
Really, probably the reason that most people didn't, I don't think we're probably not attracted to caves is because it looks good on the outside. Maybe it's hey, look, there's a shelter. It's in the side of the mountain. Looks like it won't rain there. Let's go in. There's usually something that already lives there. And whatever it is, through the musical chairs of who gets to live there, it's always the thing with the fangs. It's always the thing that ate everything else that wanted to call that cave home.
Exactly. We inherited it somewhere. Got it. I'm going to go to bed. It's bedtime.
Yeah, it appears to be the hour of the ending of the after show. After show minions, thanks for hanging out with us. We will see you again next week.
That's right. We will be back, and we hope that you will be too. Bring a friend. Bring a friend to enjoy this time of science with us next week.
Oh,
Thanks.
I got to address someone here. Yeah. Did we report a while back of scientists looking for other petite scientists to explore some bones in caves? Yeah,
Yes.
that was the Hominin, which if you Google Hominin, there is a pretty neat documentary on it. But that was one of the things that they had a very difficult, narrow entrance to the back ends of this annexes of this cave, and they needed one of the qualifications that they really wanted people who were skinny and short, but possibly bad rock climbing skills or spelunking skills combined, and we're anthropologists. It was a pretty amazingly. There's plenty of that category because a lot of people responded. Yeah, that's the homo. There's another hominid running around out there. So many
Hominids everywhere. Everywhere we look, we're going to keep finding them.
Say goodnight, Kiki.
Good night, Kiki. Say good night, Justin.
Good night, Justin.
Yeah, good night, everyone. We will see you next week. Thank you so much for joining us again for another episode of TWIS.